Hi everyone, welcome to my talk, Measuring Signals from the Stomach Non-Invasively in the Scanner Using Electrogastrogram. My name is Ignacio Rebollo from the German Institute of Human Nutrition, and I have no financial interest to declare regarding to this talk. So today I'm going to talk about first an overview of the stomach and the gastrointestinal tract, and then follow how to record the electrical rhythm of the stomach, meaning equipment, the setup, and the procedure to also analyze this data, and finish on an outlook with a couple of empirical studies and also future steps on this field. So, why is it important to think about visceral signals? So we're in the physiological fMRI signals. We can think them as a friend or as a foe. In my case, I think they are friends in the sense that the normally think about the brain and intrinsic brain activity related only to perception and cognition, but the brain task is also to keep the body alive. And these visceral signals, like the heart or the stomach, are rhythmic, and they can target cortical, subcortical, and neuromodulatory structure. And in the case of the stomach, afferent signals from the stomach can reach all major neuromodulatory structures. So they have a big potential to influence brain activity. And we have previously, we and others have shown that the rhythm of the stomach that we're going to explain you here how to measure is coupled in the brain with a gastric network, and this is to be independent from cardiac and respiratory signals, and also from movement even. So signals from the stomach could have a large influence on brain activity, and here I'm going to show you the first steps of how to measure them and how to look at them from the brain. So now going back to the gut main function, main vital function is to help us digest and absorb the food. And it does so in great coordination with the enteric nervous system that is in charge of the immune and digestive functions. These are neurons inside the smooth muscle cell, but also we have a rhythmic cells called interstitial cell that endow the gut with our electric rhythmicity in a similar way as the heart. The gut is also densely innervated via vagal and spinal fibers. The stomach is under both influence of the both branches. And actually 80% of vagal fibers are ascending, so the brain is really listening to what the gut is doing, while the spine is 50-50. I just would like to point out that the stomach is a little bit less autonomous than the rest of the gut, so it's more sensitive on what the brain is doing, but the brain is also listening to the gut. So this enteric nervous system, here we can see a cutout of a gut section, a cross section. You can see the mesenteric plexus in between the different muscle layers, and also the submucosal plexus beneath the mucosa is where the neurons are located. And the amount of neurons here are so large that are the same order of magnitude as the brain of a cat or the amount of neurons in the spinal cord. Now, also something to keep in mind is that the gut is a sensory organ and is really extended. It's one of the most vulnerable surfaces of our brain, of our body, and it's actually if we iron out all the gut, it's 100 square meters, which contrasts the 20 square meters of the skin. So this is a cartoon illustrate a little bit how long the gut actually is. But just to keep in mind, there is a large array of bacteria, toxics, hormones, nutrients, and that the gut has to sense and send this information to the brain. And just also as an idea of magnitude, the amount of receptors in the gut equal more or less the amount of cones and buttons in the retina. So the gut is really a sensory organ. Now, coming back to the stomach is a hollow pouch whose function is to receive the food that is sent through the mouth, then to absorb it, to make strong contractions and to mix it with the acid so that it's ready to be absorbed by the intestine. The proximal stomach, that is the fundus, and the corpus acts as a reservoir and as a pressure gradient that sends food down. And then the distal stomach, the corpus antrum and pylorus, is involved in mixing, grinding, and emptying of the solid food, and also mixing it with acid. And when the stomach is empty, it's contracted and corrugated, and when it's full, it's like a balloon. And this is really modulated by vagal reflexes that really control the activity of the stomach. But again, this is an active field of research, okay? So now coming back to the rhythm of the stomach, or the gastric rhythm or gastric basal rhythm. The stomach, in order to digest, so that the contractions of the stomach that are necessary to process the food are set at a certain speed. Okay, and this is one cycle every 20 seconds. Like the heart beats every second. The stomach, whenever it contracts every 20 seconds. But even when it's not contracting, it's still producing this rhythmic electrical activity that we can see over here, propagating through the stomach from a proximal to distance way. So we have faster frequencies here in the antrum, but the abdomen, in a similar way as the bones of the head filter out the EEG, well, the abdomen filters out the electrogastrogram, and we can only see this sine wave of one cycle every 20 seconds, at least with standard EEG. So what is this gastric rhythm and this interstitial cell of Cajal? The gastric rhythm is produced by these cells that I mentioned before that are called interstitial cells of Cajal. They're kind of pseudo neurons that produce intrinsically the rhythmicity and electrical rhythmicity through ion channels. And they make direct synapses with vagal sensory neurons. So this information is related to the brain in real time. And this rhythmicity is present when we cut the stomach from the spinal cord or the vagus nerve. We still are going to have this one cycle every 20-second oscillation, and even in the absence of contraction. So let me show you this in a better way. Here we have a diagram of the stomach where we have an empty stomach, or we have a few or zero contractions, but we still have the gastric rhythm present. For example, with serosal electrodes here on the skin of the stomach, we will see these slow propagating waves. With cutaneous electrodes, or the ECG, we'll see 3 cycles per minute, so 0.05 hertz activity. Now, when the stomach is full, we start seeing these contractions that are the ones that are crunching the food and mixing with the acid. And with serosal electrodes, we're going to see these slow waves that we saw, these slow propagating waves, but also ripples in charge of the smooth molecular contractions. And with cutaneous electrodes, an increase in the amplitude of the 0.05 hertz activity. So in a way, the interstitial cell of Cajal network sets the speed of the gastric rhythm, and when contractions occur that are driven by the central nervous system, they occur at the frequency set by the ICCs. But this is, of course, a simplification, but quite accurate for more circumstances. So we have to think in a way, and this is important when you're planning your experiment, in which gastric digestive state you are going to measure your subject. Is it going to be fasting when they're hungry, so the stomach is going to be small and with not a lot of signal? In the gastric accommodation phase, so when subject is still receiving food, where there are no tonic contractions, or when the stomach is empty and is really pumping and showing strong activity. So now, coming back to the electrogastrogram, these are basically electrodes over the skin of the belly, over the abdomen. And the typical signature is a peak at 0.05 hertz, so one cycle every 20 seconds in the Fourier spectrum. And we can, when we see, this is a raw signal of one nice channel, for example, this one, and you can see the gastric cycles. Here we have one gastric cycle, another gastric cycle, which are much slower than respiration and heartbeats. We can get rid of respiratory and cardiac activity with filtering to only obtain the phase, and also to reassure you that the gradient artifact of the fMRI is also removed during filtering. So this signal is so slow that when we filter out, we have no MRI artifacts. We have tried this doing outside on inside the scanner, and it's more or less the same. Now, how to record the amplifiers? First, I'm going to talk about the equipment, the amplifiers, the electrodes, the setup, the placement of the electrodes, the procedure to actually record the ECG, the duration of the recordings, some factors considered before the recordings, and a little bit about artifacts. Most of this stuff is coming from this great paper that I had the chance to collaborate with from Nikolai Warpert, a PhD postdoc, ex-PhD student from my ex-supervisor, Catherine Talon-Moudry. So if you really want to get this into, I really recommend the paper that has some MATLAB code associated with it and some example data to pre-process it, but I'm not going to cover the code on this talk. But it's quite a tutorial-like paper. So, amplifiers. There are several options to record the ECG inside a scanner. I have typically used brain products solutions, and basically any bipolar setup. I know that Biopack also has a ECG solution, but there are probably other brands out there that they can offer this solution. Basically, what we need is something that offers DC recordings, because we don't want to filter out the slow signal from the ECG. So I'm not timing this. So DC recordings are needed. And basically any EMG or ECG device, as long as it has DC recordings, will be able to do it. Also, EEG device like electroencephalography amplifier will also be able to do the job. What we'll typically do is that we'll put the electrodes on the belly of the patients. We hook in this case, the box where cables are attached, and the amplifiers at the level of the feet. But this is one possible setup. The important thing is that everything, because we are in the scanner, everything should be taped down, no cables moving, no cables lying around, no loops, right? This is like in any physiological recording. Regarding the electrodes, as I say, any standard EMG or ECG electrodes will do. I was able to record the ECG using an EEG cap, not in the scanner though, but probably it's also possible. But yeah, any standard ECG electrodes will actually do, MRI compatible, of course. Now, regarding the setup, there is no common standard for the field of gastroenterology and also for cognitive neuroscience. And one of the problems is that the position of the stomach varies across participants and even within participants throughout the day. So one possibility is to get an ultrasound, a portable ultrasound, and check where the stomach is, and then put the electrodes there. But not everybody can do this. The rationale that I'm using right now is put as many electrodes as possible, and then choose the one that has the best signal. Okay, the largest peak at 0.05 hertz. What we have previously done in the scanner is to use a grid of 4 bipolar electrodes. So this means 8 cables and a ground. And basically, we compare the activity of one electrode to another. So for this, I think this is explained on my eLife and Journal of Neuroscience paper, but the rationale is that we have to measure the distance between the umbilicus and the xiphoid process over here. And then 2 centimeters above the umbilicum, and along the mid-clavicle line, we put a first electrode and the positive derivation 4 centimeters away. Then in the 2 centimeters below the mid distance between here and here, we put another row of electrodes. In the square formed by these 2 electrodes, with 4 electrodes, we put another one, and then the last pair of electrodes on the same level here, but one on the 2 centimeters to the right and to the left of the umbilicus. But this is one possible setup. I think everybody should find one that works for them the best, and there's room for improvement for sure. Now, regarding the procedure, something to keep in mind is that have a shaver or an electrical shaver in the lab in case the subject is too hairy or hairy at all, because of course, if there is bodily hair, the electrode will stick. So the first step is to, as any electrophysiological signal actually, to rub the skin with medical alcohol and to apply abrasive gel. Here, for me, the trick is to do it until the skin is red. Always research assistant is like, they're afraid that the subject you really have to rub it for me to work. And then place the electrodes according to the setup. Check the impedance to be below 10, 25. In MRI, we can go up to 50 kiloohms. And then make sure that the subject is comfortable and that is able to stay in the same position without moving, especially coughing or anything. So note any movement that happens in recording, especially coughing and sneezing or talking. So for the ECG, typically we have long recordings. This is because the ECG signal is slow, and the stomach, unlike the heart, responds slowly to changes. So when in doubt, longer recordings are preferred, but really depends. Here, there's if you want to include the ECG in research, you really have to think why and what is the signal you're interested in. For resting state, previously we use between 10 and 15 minutes of recordings, which are 30 to 45 ECG cycles. But for behavioral studies, it really depends on the time scale of the phenomena you want to study. For example, something to keep in mind is that because of filtering, because we have to filter at such a slow frequency, we apply a bandpass filter. The end at the beginning of the MRI acquisition makes a ringing artifact. So we typically have to discard 30 seconds of brain and ECG data before and after each block. So this is something to keep in mind at the experimental design level. If you have many blocks, you have to add this buffer time. If you have long blocks, you are more likely to have artifacts. So there's a trade-off here. Some subject-related factor to consider. Typically, we don't record BMI larger than 25 because, as you may know, fat tissue is insulant, and the amplitude of the rhythm decreases with the thickness of the abdominal wall, also if it's only pure muscle. Another important thing to consider and to discuss is which level of fasting you're going to have for the recording. Complete gastric emptying takes 4 hours in a healthy subject. So some authors propose 6 hours of fasting and not drinking water, 2 hours preceding the recording. However, after long fasting, the amplitude of the rhythm drastically decreases. So here there's a decision to be made. If you are interested in the contractions of the stomach, you might have it the subject after eating. If you're interested in the more bottom-up, theoretically bottom-up, only instances of Cajal, gastric rhythm, gastric basal rhythm, let's call it, then you want them to be fasted so there are no contractions. Here, that really depends on the scientific question. So doing that before recording is important to take care of the artifact because long recording time and filtering makes that the artifact spread out, and random motion, moving arms, stuffing, turning, coughing, all this will distort the electron-skin interface. And avoid any hanging cables, tape everything around, and reschedule your subject 6. And this is some example of the artifact you will see. This is data without motion artifacts. This is when subject is reading and turning pages. This is when it's turning, when you touch the electrodes, or when they start coughing, or when they sit up and lay down. So all these will have a huge influence on the ECG. So avoid artifacts as always after recording. So I'm going to now do a little overview about the recording. So a little overview on the data analysis. The idea is that we first will downsample the signal that we acquire, because typically we acquire at 5,000 hertz, but the stomach signal is very slow, so we are not interested in that. But to get rid of the MRI artifact, we have to sample it very well, but then we can simply downsample. Then the main trick is to identify which of all the channels that we put on the subject has the largest signal at 0.05 hertz. And for this, we'll do a fast Fourier transformation to identify the frequency and the channel for that subject gastric rhythm. Then we'll simply bandpass filter that channel at gastric frequency, and then apply the Hilbert transform that we can use for getting the amplitude time series and the phase time series and use a regressor of interest. And of course, check for artifact. So this is a little idea. We have to pick which of the channel has the largest peak to obtain this phase and the amplitude, of course. So the review is the idea is to picking, that is more tricky part, let's say, is that we have to find a Gaussian-like peak in the power spectrum of at least one channel. Ideally, this peak frequency should be consistent across channels. And in many cases, if two channels have similar power, but one of them has a shoulder, meaning a peak in a slower, faster frequency, we tend to choose the one without the shoulder, so we are sure that it is only the gastric rhythm. Here is one example of a peak that is congruent across channels. So in this case, we'll choose this channel because it's the same frequency across channels. But if no consistent peak can be found across channels, or if the peak has very slow voltage, we tend to discard it. So this is a case in which it's actually not possible to know which of the channels is showing the gastric rhythm. Maybe all of them has displayed some gastric rhythm activity, but it's not clear there is only one gastric frequency for the whole stomach. So in this case, we have to discard this recording. So I'm going to give you some examples and one more step by step. Here again, we have a super clean recording with one channel, all the channels having the same frequency, but one electrode, this one over here, having the largest peak. So it's clear that we show this because it's showing the largest gastric basal rhythm activity. Of course, this is after computing the power spectrum, and typically I use a hand tapper FFT with the Welsh method. So we select the channel with the peak between 0.33 and 0.66. Here I have some MATLAB code on the paper and GitHub that like automatically detects a peak here. And here we have another example of a congruent peak. example I showed before, and then we filter around here and get the filter signal and apply the Hilbert transform now. Here is one example, another example of another case in which we have a congruent peak. Here, some other examples of cases where there is no congruent peak, or for example, here we don't know if at some point the frequency of the stomach might change, so at some point of the recording is faster and the slower. Often we discard this recording. Here again, this is not a super clear peak, so we have to discard this. Here in this scenario here, I don't know, because we have one channel with at least a lot of power, but typically, this is kind of the hard science of the ECG. It takes a lot of eye training, and there's some criterion to make it here, and it's important to be explicit about which criterion you use to discard or to keep subjects. So in a subject that has a highly regular rhythm, from the raw ECG, we filter around, we get the amplitude envelope. This is a nice fMRI regressor over the face. And it's not always possible to do this. Sometimes we get noisy channels, as I showed before, but it's possible to recover them somehow. Here, for example, in this subject, there is no clear peak between 0.33 and 0.66. And if we look at the FaceTime series, we see there are a lot of artifacts going on here, but we have a period of the recording that if we filter here, we see that there are not so many artifacts. So what we can do is that we can filter only here to find where the peak is, filter in the portion of a signal where is actually no artifacts, make an FFT there, and here we can see a consistent peak across channels. And if we filter around here, then we see that actually there are less peaks. So this is one way to recover a noisy channel. Okay, so overview of data quality check. We have to make a power spectrum and find a peak in the normal gastric range and a peak that is congruent in at least a few electrodes. That would be ideal. So this is from the paper I showed before. Across 127 subjects, 20, so 17%, we are not showing a clear, consistent, congruent peaks. So we have to discard the recording. If yes, well, we have another criterion, that is if they have 70% of cycles in normal gastric range, and if so, is a clean recording. So I'm not using this criteria a lot because it really depends on what you're doing. Normal gastric is basically the frequency between 0.03, 0.03 and 0.06. The stomach can also show faster and slower activity, so tachygastria and bradygastria. And this might be relevant for emotion. So it really depends on which kind of study I'm doing. And here I'm going to show you an example of the first study that I think used fMRI and ECG. So first, the fact that we know that changes in the frequency and power of the ECG responds to emotional clips and music. And Harrison and Critchley in Journal of Neuroscience found that regions activated by this gas were also overlapping in the anterior insula with regions overlapping with tachygastria, so faster stomach activity. So discarding cycles only because they're faster or slower than normogastria, it not always makes sense. Also, something also to keep in mind is that typically this is a standard procedure in which we use one electrode. At the end of the day, we only use one electrode. But if you do a CT scan, we found out that the stomach position might not be covered by that electrode. And in those cases, we are going to think that the subject doesn't have a normal gastric range, but actually we are missing the stomach. So what the field is doing a little bit right now is put an array of electrodes with a lot of them, so up to 50 electrodes, let's say. And here we see that actually we have what we thought it was an unhealthy stomach was also a healthy stomach. And also we can extract measures that are much more correlated with symptoms that the standard measures are not. So this is something also to keep in mind. Another possibility, and this is what I typically do, is I only work on the face so far on the ECG, and try to study MRI brain phase synchronization. So for this, across 60 participants, we record resting state and fMRI ECG in participants that fasted for 2 hours, and we compute the phase synchronization using phase-looking values. So this is an amplitude that completely ignores amplitude and directionality. And basically looks how consistent is the time delay between the two signals across time. Here you can see a voxel that is consistent with the stomach and a voxel that is not. And for this, we use this equation. And something important to keep in mind, that's also why maybe I mentioned this study right now, is that you have to have a chance condition, right? Typically what we use here is that either we compare this empirical PLV map with a chance PLV map that we can obtain either by shifting in time the ECG or by using the ECG of other participants as a surrogate measure. And then we use a t-test across these two conditions. And we got this gastric network that is spread across somatomotor and visual networks, but also some cortical structures like the striatum, thalamus, and cerebellum. And right now we're trying to replicate this across a larger cohort of participants. So to conclude, ECG has a lot of potential to understand brain-body interactions in cognitive neuroscience. However, careful planning and experimental design and setup is needed. Feeding time, like when the subject is going to be fed with respect to the scanning and which digestive state we're going to aim is also important. And the timings of the condition, make sure to have enough time and to pilot the data to make sure that the ECG has enough time to change for your stimuli. And in more general, I think like the field is moving for causal manipulations of the stomach and of more refined measures of the stomach, such as invasive or high-resolution mappings of the stomach. I would like to thank my collaborators and you for your attention, and my funders. And yes, if you have any questions, you can reach me by email or Twitter if still a thing in a couple of months. Thank you very much for your attention. I wish you a great day.